Why study sex difference? It's more than just sort of interesting. Every cell has a sex, and sex influences fundamental biology through hormones, estrogens, testosterone, through X chromosome dose, through the presence or absence of a Y. And true sex differences in aging exist. For example, women live longer than men in aging and with diseases like Alzheimer's. And most importantly, if we decipher what makes one sex more resilient or vulnerable, we can unravel new therapeutic pathways to treat both sexes. So can we find new therapies, new molecules, new therapeutic pathways based on sex-based resilience? This is a really striking graph from the United Nations from data collected in 2021 with the life expectancy of women on the y-axis and of men on the x-axis. And what you can see is that in every society that records mortality around the world, from Somalia to Japan, women will live longer than men. This is true in very extreme environmental conditions, like in famines and epidemics. Women will live longer than men. Girls will live longer than boys. This is also seen in much of the animal kingdom, whales and lions, in bears, in many great apes, even in many strains of mice. The females will live longer than the males, and certainly there must be a societal, environmental, and cultural contribution, war, increased risk-taking by males, but there also is a biologic contribution, and that's what we are really digging in. And for example, in a situation where we remove society and we culture brain cells, the XX cells will live longer than the XY cells, devoid of culture and society. Now, the first question we ask is, do mice recapitulate human sex differences in lifespan? And one of my graduate students, Emily Davis, aged out mice for several years. This is a lifespan graph, and found indeed that female mice, XX mice with ovaries in red here, will live longer than the male mice in just normal aging. Now, when one sees a sex difference, how do you dig into that? What are the mechanisms of that sex difference? How do you discern and dissect causal mechanisms of sex difference in longevity? So Emily asked, do sex chromosomes, different between males and females, XX versus XY, or do gonads, ovaries versus testes, contribute to increased female lifespan? And she used this really unique model called the 4-core genotype model to attack that question. In this model, the testes determining factor SRY is knocked off of the Y chromosome and onto an autosome, so it can now be inherited by males or females. And this model produces 4 sex genotypes. So put your Sudoku caps on, I'm gonna ask you a question. We make XX mice with ovaries. Those are the typical females. We might make XY mice with testes, typical males. And then we produce XY mice with ovaries. And these mice are female. They show female reproductive behaviors. They have estrogen, progesterone, they have reproductive cycles. They're female mice. They only have XY chromosomes. And then we produce XX mice with testes. These are male mice. They have male reproductive genitalia. They fight in their cages. They produce low levels of sperm. They're not reproductively fertile. But this model can give us very powerful data. So if, for example, the mice that are circled here, XX with ovaries and XX with testes, if those mice lived longer than the mice that are underlined, XY with testes, XY with ovaries, what could we conclude about a causal contribution to longevity? I should warn you, I love long periods of silence. Someone said louder. The question is this. The question is this, is that if the mice that are circled live longer than the mice that are underlined, mice that are XX with ovaries or testes live longer than the XY mice with ovaries or testes, what would we conclude about a causal contribution to longevity? Okay, right. And so many people said this also, but we would say that there's something about sex chromosomes that's contributing to female longevity. That's one outcome, but there are many outcomes of this model. It could be that mice with ovaries live longer than mice with testes. We follow the science, but again, really powerful models that give us insight into causal contributions. What we found was a very striking sex chromosome effect. That is, the mice that are XX, whether they grow up with ovaries or testes didn't matter. They live longer than the mice that are XY, growing up with ovaries or testes. Really powerful data. Now, we're very interested in understanding what are the things that help us to live longer, and can they help us to live better? And that is the reason we're studying female longevity. But every organ, as we've heard, can age differently, and females live longer. They're also more frail in terms of bone health and muscle health, but the brain is a different story in typical aging. So, is there biologic and molecular evidence for slower brain aging in women? Again, we're not talking about Alzheimer's or Parkinson's disease. We're talking about typical aging minus dementia. And in one measure of the epigenetic clock, Steve Horvath reported many years ago, when he looked at several regions of the brain, that female brains, actually across the lifespan, aged slower than male brains. Others have shown using PET imaging, glucose metabolism of the brain, that the aging female brain is metabolically younger than the male aging brain by approximately 4 years. And this is again seen across the lifespan of adulthood. Now, what about cognitive aging? Cognition is one of our most valued output of the brain that we really strive to preserve in our goal for healthy aging and for health span. Do women show differences in cognitive aging compared to men? They do. Again, in the absence of dementia, women will show less cognitive aging in many populations of clinically normal adults, excluding those with dementia. So you can see from this graph, which is from the Mayo Clinic, of a couple of thousand individuals, that women in orange will show slower cognitive decline, worse memories on the y-axis, but they'll show slower cognitive decline compared to men. We've seen this at our own Memory and Aging Center at UCSF. It's also been shown in the Baltimore Longitudinal Study of Aging. And other studies that include dementia will show you something different, where women decline in a more steep fashion. But in typical aging, devoid of dementia, females tend to show less cognitive decline. Again, why do we care? Because understanding what confers resilience in one sex could reveal new targets for treating both sexes. All right.
Is there any data showing cognitive maturing in the early age, early childhood?
It depends on what type of cognition, our ability to think, judge, hold memories, use language, and that really is different between girls and boys early on. In many domains, the girls will mature faster than the boys in adolescence. Yeah. What about mice? Again, these are the models that give us really powerful insights into mechanisms. My graduate student, Francesca Marino, used a test called the large Y maze to study spatial and working memory, navigating where one is in space and remembering in the short term what one just did and what one just experienced. And the details of the maze don't matter, but on the y-axis is essentially memory. 1 means no memory. On the y-axis, a higher level means better memory. And what you can see is that in young mice, XX and XY, there's pretty robust memory, as expected, spatial and working memory. With aging, both sexes show a decline, but that the male mice show a more marked decline compared to the female. She also used this 4-core genotype model that we just thought through. Another model is coming up, and asked whether it was sex chromosomes or gonads that contribute. There were many interesting findings. I'll show you one of them, that in mice that grew up with testes, the XX mice showed better cognition than XY. So essentially, adding XX to individuals with testes improved their cognition, telling us, again, a very important contribution of sex chromosomes. Now, when we understand that there is a sex chromosome effect, the next question arises, is it the second X that's helpful, or is it the Y that's detrimental? This is where the next model comes in. So Francesca asks, does the second X chromosome increase cognition, or does the presence of a Y chromosome decrease? So this is called the XY star model, and it produces 4 different sex genotypes: XX, XY, 1X, that's XO, or XXY. So I'm going to ask you another question. What if the mice that are circled, XX and XXY, showed better cognition and aging compared to XY and XO? What could you conclude about the contribution of X versus the contribution of Y? So the circled mice do better than the underlined mice. Okay. Right. So in that scenario, and there are many different scenarios, but in that scenario, in this scenario, you could conclude that the second X was beneficial. But what did Francesca find? She found just what you predicted. That is, or what you concluded from the last slide, that is, that the mice with 2 X's, whether they were XX or XXY, showed better cognition in aging compared to the mice with 1X. Really powerful and clear data that that second X chromosome improved cognition in aging. If we break down the different experimental groups, you can see that adding an X to XY, aging males, improved cognition. Taking away an X from aging females decreased cognition. So in a very tractable way, we've demonstrated that there's something about that second X. So let's talk about the second X. It's a major source of sex difference. We know now that it contributes to female longevity and cognitive resilience, but how is completely unclear. The X chromosome contains 5% of our genome, and it is enriched for brain-related genes. So there's more, there's more brain-related genes on the X chromosome than there is on any other autosome in our genome. And in mammalian biology, and that includes human biology, 1X is inactivated during development. Around the 5th or 6th day after fertilization, one of the X's will shut down randomly. And that's called X chromosome inactivation. So here's a puzzle. If 2 X's are better or matter in longevity and cognitive resilience, how can that come to be if one of the X's is shut down? So males will have 1 active X and females here have 1 active X. So why would that second X matter? That's what we will discuss now. And again, a really ripe area that I believe will have a lot of new therapeutic targets for brain resilience. So the first scenario is baseline X escape. As you recall, in development, one of the X's shuts down, but maybe it's not totally shut down. So despite that formation of the Barr body and lionization of the second X, there are still factors that will escape transcription. One of those is KDM6A, a histone demethylase. It unwinds the DNA and opens it up to more gene transcription. And I wanted to show you, I just wanted to take you like deep into the lab under a microscope to show you what X chromosome inactivation escape looks like. So this is the nucleus of a mouse neuron in blue. And we performed FISH, which is fluorescence immuno in situ hybridization. And what you can see in pink here is 2 dots. And that's transcription off of the active X chromosome. This is the one that's active, and this is the one that's inactive. Exist is coding it to make it inactive, but you can see that KDM6A is being produced at 2 sites in the nucleus. That tells you that there is baseline X escape. In the male nucleus, there's 1 site because there's 1 active X, and KDM6A is being produced off of that active X. So really just a beautiful demonstration of escape from that X inactivation. Now, there are select factors that escape from both humans and in mice. KDM6A is one of them, and we tested the idea that KDM6A is lower in males than it is in females. But what if we, through genetic engineering methods with lentivirus, what if we boosted up KDM6A in males to female levels? That's what we did here. And what we found in a memory test using, in this case, Alzheimer's, we've also done this with aging mice, we see that with aging or Alzheimer's, in this case, this is a water maze study, the memory gets worse in male mice. But if we bump up KDM6A levels, again, this factor that unwinds the DNA, that memory normalizes essentially to youthful or non-Alzheimer's disease levels. So bumping up KDM6A, this X factor in males, boosts their cognition. What about, you always ask, well, what about the human condition? Because we're really interested in mice, but ultimately none of this matters if it can't translate into humans. So we worked with the neurogeneticists, Jennifer Yokoyama and Luc Bonham, and identified a genetic variant of KDM6A that associates with higher levels in the brain. So males and females, if you have this code of KDM6A, this specific code, then you will have higher KDM6A levels in the brain. And those individuals with 2 copies of that genetic variant here in blue did not show cognitive decline over a decade in a cohort called ADNI, which is individuals that are normal and at risk for Alzheimer's disease. Without that variant, people showed pretty significant cognitive decline. And this gave us essentially human evidence that KDM6A is important in brain health and may be very important in preserving brain health. Now let's talk about another way that this is the second example of how the second X chromosome might be important in brain resilience. And that is the idea that X is silent, right, in development, but maybe in aging, there's an awakening of that second X. That was Casey's hypothesis, another graduate student in the lab. She hypothesized that while this inactive X is silent in youth, that it awakens in aging, and maybe that helps with female longevity. Maybe that helps with cognitive resilience. And so through a very unique genetic model, we won't go through the details, but she developed a way to tell if if a factor was coming off of the active X or the inactive X. And what she found in this map of gene expression in different cell types in the hippocampus is that in aging, that there were certain genes that essentially turned on from the silent X. So this is a map of genes from the silent X, and you can see in young, there is no expression. In aging, shown in green and yellow, there is an onset of robust expression, indicating, giving us clear evidence that that second X is awakening in aging. And it awakens in non-neuronal cells, including astrocytes, microglia, and oligodendrocytes. Now, this is the third, and I think the most interesting example of how a second X could confer resilience. And that is on mixed parent of X origin. So going back to really basic biology, an inheritance of the X chromosome. Males will inherit their X chromosome from which parent? From mom, always from mom. And the Y is from dad, always from dad. In females, the story is different. There's more of a mix. So X can come from mom and X can come from dad. And that's what's shown here in this figure. M is maternal, P is paternal. So in one cell, there will be mom's X active, and in the adjacent cell, there can be dad's X active because of random X inactivation. And this makes for beautiful biology. This is from the hippocampus, the area of learning and memory in a mouse. We would also see this in human. This phenomenon is preserved in human. And these are all cells in a region of the hippocampus. And they're fluorescently labeled. So if the cell shows mom's X, it's green. If it shows dad's X, it's pink. And wow, what a mosaic. Side by side, you can see that even though these are the same types of cell, they have genetically identical X's, each cell will differentially show mom or dad's X. Even in a litter from the same mom born at the same time, that pattern of whether mom's X is dominant or dad's X is dominant or they're equal is very different from pup to pup and even from human to human. So in this little mouse, mom's X is predominant. In this one, dad's X is predominant, and they're sisters from the same womb. This is also true in humans. I might be 80% mom's X, and you might be 80% dad's X. And at this time in human profiling or therapeutics, there's really not an understanding of an individual X chromosome expression, of a female individual's X chromosome expression. And I'll tell you why I think this could make a big difference in personalized health for women. Okay, so Samira asked the question, she's a postdoctoral fellow in the lab, does that mosaicism in females help in aging? And actually, just to ask a basic question, are cells that show mom's X different than those that show dad's X? And to do this, she used a really neat genetic model, again, where she marked chromosomes. Neurons, these are neurons in the hippocampus. Neurons showing mom's X are green, showing dad's X are red. So she just separated out these cells and asked, are they different? Again, they're genetically identical X's, but are these cells actually different? And through a rigorous experimental approach where she separated out the cells, she found something absolutely remarkable and unknown to us. And that is that there's a really big difference between these two neurons. Those that show mom's X will silence multiple genes, will silence multiple factors. Those with dad's X, those factors are robustly expressed, demonstrated here. So mom's X will actually silence off several important genes in brain function. Mom's X imprints or silences genes in both the young and the aging brain. This persists into aging. So, well, that's interesting, but does it matter for the brain? She looked at cells that were isolated from the brain, and she examined their properties in a dish. And she looked at whether maternally expressing neurons showed different properties in neural functions. So she first started by making a really unique mouse model. This is the normal mouse with the mosaicism of mom's X and dad's X. And then she made a mouse, a female mouse, that only shows mom's X. And this would be similar to male genetics, but she made a female mouse that only shows mom's X. And she looked at how the brain cells connect with each other. And she found that mom's X, that neurons showing mom's X, will decrease the complexity of neurite outgrowth, of brain cell connections. What about cognition? Again, our most valued output of the brain. She found, in fact, and we'll focus here on the right side of the graph, that in mice, those mice, that female mice that only show mom's X, that there was an increasing worsening of memory with age compared to the mosaic female mice. So that a direct demonstration that mom's X worsened cognition in aging females. Now, because this was seen increasingly with aging, she wondered whether the maternal X can accelerate brain aging. We've heard some about epigenetic clocks or measures of biologic aging. This is one of those clocks, the Horvath clock. And what Samira found is that female mice with only mom's X show accelerated brain aging compared to the mosaic mice. And that if she looked within the same brain and separated out neurons, the brain cells that show mom's X from those that show dad's X, that the neurons showing mom's X showed nearly double, if not more, of accelerated biologic aging compared to cells showing dad's X. Very, very clear and fascinating biology. So in summary, what I've shown you is that women live longer than men, and they often show cognitive resilience in aging, and that sex chromosomes have something to do with this in a causal manner. Specifically, the second X is probably a treasure trove of aging therapeutics, cognitive therapeutics. And that very provocatively, that the maternal X chromosome, the one that we inherit from our moms, blame your mom, will silence putative cognitive loci and actually impair cognition and its substrates in aging, not in youth, but in aging. And this is important at the end of the day, because if we can understand mechanisms of X chromosome function in the aging brain, again, there may be new therapeutics that don't have to do with feminization or masculinization, but may be very specific cognitive therapeutics. Another reason this is very provocative is that I haven't shown you sex differences between what happens in a male versus a female in terms of parent of sex origin, but it suggests that if males only have mom's X, that there may be vulnerability to aging or to other cognitive problems in later in life. It also means, again, specifically for women's health, that if Anastasia is just stochastically 70% dad's X, maybe she is more protected from cognitive aging compared to me. because I'm more mom's X. Maybe this could help to predict in a diagnostic way vulnerability to cognitive aging or vulnerability to other diseases. We focused on the brain, but you can imagine what this could imply for different organs of the brain, maybe different health trajectories and maybe different disease vulnerabilities could be identified by understanding X chromosome expression in the female. These are the folks who did the work and my sources of funding. And thanks for your attention.
So we have time for a few questions.
Great. That was a really exciting talk. Thank you. So of all of the exciting data you showed, I think the one with the single biggest effect size was the very last thing you showed, where you had this enormous difference between the paternal and maternal. So you didn't explain what your genetic model was to do the skewing, but can you skew it the other way? Can you make females that only inactivate the maternal one?
Mm-hmm. The genetic model for skewing is a selective deletion of X-cyst from an X. X-cyst is the long non-coding RNA that shuts down the X when it's expressed. So if you delete it from an X, then it is automatically the active X, and by default, the other one becomes inactive. We have tried for years to skew it in the other direction, and we have failed over and over and over again, based on the very basic biology that in the developing embryo, that dad's X needs to be, that one of the X's needs to be silenced, and that skewing it the other way messes with the biology such that the fetus can't form if we try and make dad's X active. Dad's X has to be silenced in extra embryonic tissues. So we have failed, but we are getting around that by separating out maternal, different cell types that are maternally derived versus paternally derived within the same brain using fluorescent markers. But we really wish we could have done that study.
Dana here.
Yes, hi, Tom.
Really fascinating. So could you imagine that there are instances where over generations only the maternal or the paternal is inherited? And second sort of question is there any evolutionary purpose behind this?
Oh,
Simply stochastic.
Yeah. So remind me what your first question was. I'm maternal X active dominant.
Whether you can
generations. So we don't know the answer to that. My speculation is that the epigenetics are wiped at each generation, but it hasn't been tested to know whether, if you're consistently getting mom's X down from generations, whether that could be detrimental, or whether maybe these centenarian super ager females that live to be 115 and say, I may be very old, but I'm not an idiot. Ranjan told us that fun story. But maybe those women are predominantly dad's X and have been for generations. Who knows? I think this is a very exciting area. We just don't know. And then remind me your second question. Evolutionary. What would be the? I would love to think with you all on that. I do not know what would be. I think it may be. I don't know. Yeah, I could speculate wildly, but I just don't know. I think that we've just opened an area we hope that others will study, and we hope that ultimately gets integrated into women's health and diagnostics.
We'll do one more question and we'll move into the panel.
Yeah, so I just wanted to piggyback maybe on, on Aubrey's question.
Oh.
So do you know if it's a mom's X versus dad's X question, or if it's a primary X versus backup? In other words, do you know if it's a difference between the chromosomes, or just, your mom's X is used throughout life, and then you switch over to dad's X, which has not had the wear and tear of being used throughout life and confers some more resilience?
From what we know from X chromosome biology, there is no switching in the brain, at least. There might be some in blood cells, but once a cell is fated to be mom's X expression, then that is the way for for the rest of the life. And then and then we show data that other cell, that the other X might kick in later in aging.
Right.
Whether that's mom's X or dad's X, or whether that makes a difference, we don't know.
And that happens in the brain as well, or just in other parts of the body, not in the brain?
With the other X kicking in, we have demonstrated it for the first time in the brain, and it has not been systematically, to my knowledge, looked at in other parts of the body.
All right.
Have you tried looking at 2 copies of the same X in all of your animal models rather than having 2 different X's in each animal?
Ask me another way. I don't understand your question.
Yeah, so I'm not sure if this would be possible, but I mean, obviously you want varied inheritance in each animal, but if there was some way to control on the X to have 2 copies of the
From
exact
mom.
same X for
Yeah.
every animal, yeah.
Yeah, we haven't figured out how to do that. I'm not sure it's possible, but I probably in the future there is a way to do that. We haven't done it yet, but it's a really neat study. You might be able to see like a double dose of mom's toxicity, or a double dose of dad's resilience if one is able to.
Yeah, I just
that.
think it would really control for the method that you used to enact X.
Although all the very last data that I showed you, mom's X versus dad's X, with really striking, again, this is brain. In brain, we get excited about 20% changes. And in this data are neurons from the same brain without any kind of genetic modification. So these are the neurons showing mom's X that are just adjacent to the neurons showing dad's X. And they show very, very clear accelerated biologic aging without any genetic manipulation. These are the mice that are genetically manipulated. But that's why this was so important, that in the natural condition, there is such a big difference between parent of origin.
All right, join me in thanking Dana
Thank you.
for a very insightful talk. We'll transition into a panel, or I'll stay outside.
Okay.
That way. So the way I'm going to run this is before we start, I think there was an interest in understanding the audience makeup. And I know like we went through applications and every one of you applied to be here. For our panelists and for myself, we're going to do kind of a quick show of hands. I'm very interested to know how many of you are academics. Please raise your hand. You work in a research lab. You are interested in the longevity space as an academician. Okay. What about you're an investor or you work in the investment ecosystem? Okay, awesome. And then lastly, our startup folks, who's doing or building something in the startup domain? Probably multiple hands and multiple fronts. So, okay. Now this is helpful. I think for us, it's helpful to understand so we can frame the questions and kind of think about our audience makeup and such. So with that, I mean, we've heard a lot so far about aging, longevity, the brain, kind of thinking about more broadly what that does to impact our health and kind of living longer. And if we were to live to 100, what would that look like? So I'd like to open this up to start to think about what are the most promising new therapeutics that you're thinking about or have seen that are working on